All right, we good to go ahead and at least get my nonsense out of the way.
Yeah, we'll get started.
All right, and I'll try to keep this brief. I just counted, and based on the bubbles at the top of the slides, it looks like we're in for 45 slides. So we've got a lot of material to cover. So it's my pleasure to introduce today's seminar speaker, Mike Hogan. So Mike is a PhD candidate in my lab. He joined the E&E program in 2016. So when he joined the lab, I think the idea was that he was going to work on snake venoms. But ultimately, as you'll see in the talk today, he kind of went off in a different direction to study the evolution of sensory systems, mostly in rattlesnakes. And so, genetically, when we're studying venoms, these actually can be pretty challenging to work with. So the venom genes themselves, they exist in these really, really large complex gene families. They have tons of paralogs, lots of copy number variations. So really challenging to work with in terms of doing de novo assembly, identifying the genes, studying their molecular evolution. Apparently, Mike saw all the trouble we were dealing with in venom genes and decided to try to turn that up to 11. So he decided to work with these giant olfactory receptor genes that exist in gene families that are 10 times at least as big as anything we ever see in venom gene families. So it's a ridiculously challenging system, but as you'll see, he's been making really outstanding progress on this. So in support of his work this last year, Mike received a really prestigious evolutionary, ecological and conservation genomics research award from the American Genetic Association to support the work on sensory evolution he's been doing. He got his first chapter of his dissertation published in the journal Molecular Evolution, which also includes the first published genome of his study species, the eastern diamondback rattlesnake. He's also been a co-author on 6 other papers from the lab, and he's got some really good, really big papers in the works right now, which is going to take up most of what he's going to talk about today. So without any further delay, I'll turn things over to Mike to talk about molecular evolution of sensory perception in rattlesnakes.
All right, can everyone hear me? Cool. All right, well, thanks for that introduction, Darren, and thanks for everyone for joining us today. Today, I'll be discussing my PhD dissertation research on the molecular evolution of sensory perception in rattlesnakes. Predatory traits are traits that enable one organism to consume another. For example, courtesy of FSU's very own Dr. Greg Erickson, bite force is a trait that dictates diet in crocodilians. Traits like this are directly tied to fitness and are generally easy for biologists to identify, but functional overlap is not uncommon. Since all traits are, to a large extent, products of genes and gene sequences evolve in predictable ways, we can reconstruct the timing of events that led to the emergence or optimization of predatory traits. For example, the evolution of keratin genes in birds closely reflects lifestyle shifts and the usage of talons for predation. Pit vipers, the group of venomous snakes that include rattlesnakes, are often recognized as masters of predation, thanks to their impressive venom system. This includes foldable hypodermic fangs, lightning-fast strikes, and highly toxic venom. As ectotherms, they use minimal energy and could go long periods of time in between meals. However, I would argue that pit vipers are more so masters of perception. After all, the first step in predation is perceiving your prey. Adaptive traits experience selection towards functional optimums, and this is in part driven by the coevolution of predator avoidance and prey. Sensory perception must work for venom to evolve, and I like to illustrate this by saying, if you can't find it, you can't kill it, and you can't eat it, since failure at each point leads to the same fitness response, a hungry snake. So to summarize my research, my initial goal was to determine what genes allow rattlesnakes to detect their prey. From there, I investigated if there was a molecular evolutionary baseline for sensory perception. And finally, I tried to figure out if genetic integration between sensory perception and venom was evident. In other words, I looked for molecular support for the coevolution of these two traits. Sensory genetics is a fairly new area of research, especially in non-model systems. That being said, vision is one area that has received attention, so much so that we can actually predict peak spectral sensitivity directly from gene coding sequences. So in other words, we can take a drop of blood, pull the DNA from it, and predict what colors that animal can see. This has actually helped us to understand how fish evolved at different ocean depths based on light environment. Additionally, we can use sensory gene evolution to understand sensory trade-offs. For example, when cetaceans shifted into an aquatic lifestyle, they experienced trade-offs corresponding with echolocation, as well as a loss of olfactory genes. This has also been described in sea snakes, since airborne cues are not much use if you live underwater. My research revolves around the eastern diamondback rattlesnake, Crotalus adamantius. It is the largest species of rattlesnake. It can be found in the southeastern United States. It eats rodents and other endotherms. And it has 4 primary senses: sight, thermal sensing, touch, and chemoperception. So what are the genes that produce these sensory phenotypes? Let's start by covering what we already know from the literature. We know that rattlesnakes have 3 functional photoreceptors, making them trichromatic, just like you and me. These include the short wavelength opsin, the long wavelength opsin, and rhodopsin. They also have 2 main heat detecting genes expressed in their facial pit organs called TRPA1 and TRPV1, which code for cation channels. These are believed to promote the detection of endothermic prey even in complete darkness. We can also approximate their mechanosensory ability with around 14-ish genes, which is expected to be fairly consistent across vertebrates. However, we have no idea what kind or how many genes make up their chemosensory repertoire. What we do know is that chemoreceptors should be expressed in both the olfactory bulb up here and the vomeronasal organ right here, both shown in purple. And these red dots are meant to illustrate detection of potential scent cues. In order to characterize these genes, I needed a high resolution genome. Luckily, I wasn't the only person in my research group in need of a quality genome. So my project initially overlapped with a huge collaborative effort to generate a chromosome resolution genome assembly for the eastern diamondback rattlesnake. Chromosomes were recovered thanks to high-C sequencing in collaboration with the Fraser Lab. The sex chromosomes were also identified using read ratio mapping between the sexes. And altogether, the resulting genome represents the best snake genome that has been generated to date by several orders of magnitude. As a way to check agreement between our rattlesnake genome and the next best published for the prairie rattlesnake, we checked chromosome positioning of conserved genes and found almost perfect agreement across the board. To orient you to this plot, each of the segments around the outside are chromosomes, shown in white for the eastern diamondback and gray for the prairie rattlesnake. And the autosomes are ordered by size, largest to smallest from top to bottom. And I put the sex chromosomes down at the very bottom. Conserved orthologs are linked by colored lines, and these colors correspond to the eastern diamondback chromosome placement. Basically, the fact that we can see clean bands of color rather than scattered chaos tells us that there is clear agreement between these 2 genomes. We also confirmed that our W chromosome assignment appears to be correct, making it the first snake genome to fully recover the W chromosome. This was only possible because our genome animal was a female while the other represents a male. Females are the heterogametic sex in snakes, which is opposite in humans. Since nothing from the male genome matches to it, we can be confident in our W chromosome assignment. All right, so now I have a super badass rattlesnake genome. Now what? Well, a draft genome by itself is actually pretty useless. It's really just a long list of A's, T's, G's, and C's, and it's kind of like having a map of the country without any roads on it. Not very useful. If we want to get into the good stuff, we first need to find all the genes in the genome, and then figure out what parts of those genes code instructions for making proteins, otherwise called coding sequences. This was done using a combination of homology searches, RNA-seq mapping, and machine learning. I also checked each venom and sensory gene individually and identified all potential transcription factors using a machine learning pipeline. I'll discuss the transcription factors a little bit more later. From the resulting genome annotations, I identified two main families of G protein coupled receptor that were abundant and widespread across the genome, as you can see here by the different colors representing different chromosomes. The first of these were the olfactory receptors, with almost 500 individual genes identified, including 300 that were detected on the Z sex chromosome. And the second group were the type 2 vomeronasal receptors, with almost 700 genes identified, and also around 300 copies detected on the Z chromosome. Just to give an idea of what these receptors actually look like, here are some very basic 2D representations of these proteins bound to a cell membrane. Right away, you can see that the olfactory receptors are much smaller than the V2Rs and are believed to form monomers, which means one copy of the protein results in a functional receptor. V2Rs, on the other hand, are predicted to form dimers, where two copies join together to form the functional receptor. The important thing here to take away is that both of these receptors have regions inside and outside of the cell, and the extracellular regions are far more likely to interact with external stimuli, such as scent molecules, which I illustrated here in red. Looking at the gene locations along each chromosome, I found that chemosensory genes mostly occur in tandem repeat arrays, which just means that multiple copies of a certain type are found clustered together. In this figure, I colored the olfactory receptor genes orange and the V2R genes pink. I also zoomed in on one of these V2R gene arrays to illustrate what they actually look like. This dense clustering is important from a genomic architecture standpoint, since duplication events at these regions could have major effects on the sensory perception phenotype. So to wrap up the genome part of my talk, I decided to compare gene tallies between our rattlesnake genome and the human genome. Before I go on, let me just clarify that no, we don't actually have Darren's whole genome sequenced, at least not that I'm aware of. All I did was download one of the several open source human genomes that are available online, and then I used it to tally up the genes by category. In total, we found 21,596 genes in the Eastern Diamondback genome, which was surprisingly close to the 21,306 genes reported in the human genome. I also found comparable numbers of transcription factor genes between these two genomes. And I found considerably more chemoreceptor genes in the rattlesnake genome compared to the human genome. Okay, so now I've identified over 1,000 chemosensory genes. What's next? Well, I decided to investigate gene diversity of both olfactory receptors and V2Rs using phylogenetics and functional protein inference. I did this by building maximum likelihood gene trees for each of these two gene families. I then tested for directional selection across each group using DN-DS ratios, and these results were later evaluated based on functional protein regions. As a final confirmation that chemoreceptor repertoires are evolutionarily relevant, I characterized a chemoreceptor expansion in squamates by comparing gene synteny for one of the many V2R gene arrays. To quickly review, this is how DNDS ratio tests actually work. Here I have two nucleotide sequences with the translated amino acid sequences underneath. The first DNA mutation on the left side also changes the translated amino acid sequence from an L to an H. So we call this a non-synonymous mutation. The second mutation on the right side does not change the amino acid sequence, so we call this a synonymous mutation. When we see more non-synonymous mutations than synonymous, we call that positive or diversifying selection. And this signifies changing the protein, changing the structure, and potentially changing the function. When we see proportionally more synonymous mutations, we call it negative or purifying selection, generally meaning if it ain't broken, don't fix it. This is a really useful tool when comparing genes between species, but as you're about to see, it can also be used as a tool for understanding variation in the gene repertoire of a single species. For my DNDS analysis across all olfactory receptors, and again, for the V2Rs in the Eastern Diamondback, I discovered significantly more positive selection in the extracellular regions of each type of receptor. These regions are most likely the scent binding areas of these receptors. This strongly suggests that the huge gene diversity we are seeing is in fact tied to the perception of diverse stimuli. In this slide, I'm showing the olfactory receptor gene tree with the branches experiencing significant extracellular positive selection colored in green. And we can see a large cluster of these green branches on the Z chromosome marked with a yellow star. I'll talk more about that in a bit, but it's right here if you can't see it. Okay, for the synteny analysis, and by the way, gene synteny is just a way to compare order and arrangement of genes between multiple genomes. I found major V2R expansions in snakes, and this was based on gene homology traced back to lizard V2Rs. I knew I was comparing the same gene array in each of these species thanks to three highly conserved immunoglobulin regions that split up this array, which are highlighted by these giant light blue arrows. So that's here, here, and then here. Snakes evolved from a lizard ancestor, and that common ancestor likely looked more like a forked-tongued Komodo dragon than a green anole. The shading corresponds with the gene phylogeny for this array, shown on the right side over here, with the lizard V2Rs in red and orange. The ones that are not shaded are unique to snakes. Basically, the main takeaway from this is that a V2R gene expansion occurred in snakes, likely following the divergence from a lizard-like ancestor. All right, so now that I have my genes of interest characterized, I can start looking at how they are expressed. One thing to note, I will only be talking about olfactory receptors expressed in the olfactory bulb moving forward. This is partially because recovering the vomeronasal nasal organ has been too inconsistent for these types of analyses. However, I do plan to repeat these tests on the vomeronasal nasal organ in the future when I can recover these tissues more consistently. So now I'll look at gene expression to test olfactory receptors for age and for sex bias, which may be able to tell us something about the biological function of these genes. By the way, if gene expression is a new term for you, this is all I'm talking about. I'm just counting up the RNAs and seeing if one group makes more of a particular gene than another group. Just to provide some biological context, I'll read these two excerpts from the Biology of the Pit Vipers textbook. The first one is observations during fall of free-ranging timber rattlesnakes in New Jersey revealed that neonates remain close to, if not in contact with, adult males and females. This suggests that the neonates follow the trails of adults to overwintering sites. So this passage shows support for an age-biased perception with the juveniles potentially needing to detect different scent cues than the adults. And on to the second passage. Throughout most of, throughout most of the year, male and female Crotalus adamantius made similar movements, rarely over 150 meters. However, during mating season, from mid-August to late September, males traverse distances of up to 1 kilometer or more before coming to rest next to a solitary female. This passage shows support for sex bias perception, with males and females displaying different mate detecting behaviors. And actually, I'm pretty sure this passage came from some of Bruce Means's work back in the early 90s, who might be here. So hi, Bruce, if you're here, and thank you. Okay, so what exactly did I do? I evaluated RNA from 6 Crotalus adamantius olfactory bulbs and found gene expression biased for age and for sex. I also detected transcription factors, which were biased for age and for sex. And just for the record, this is my super basic definition of what I'm calling a transcription factor. It's just a protein that could swing back and influence the expression of other genes, or even itself, or even other transcription factors. In general, this regulatory stuff can get really confusing. Prepare yourself, because you're about to see a lot of these heat plots. The plots illustrate differential gene expression, where each row is a gene and the columns are individuals. All of my heat plots have the individuals ordered by snout vent length, shown down here at the bottom. So the far left is the smallest juvenile, and the far right is the largest male. At the top, you can see that the juveniles and the adults are colored blue and red, respectively. The cell colors are kind of like fire, with the warmer colors meaning more gene expression and the darker colors meaning less. From the age bias tests, I found 7 differentially expressed olfactory receptors that were all higher in adults, and 10 differentially expressed transcription factors, with all but 1 that were higher in adults. For the sex bias test, I found a strong signal for male biased olfaction. 36 of the 39 male biased olfactory receptors were also from the Z chromosome, supporting sex chromosome influence in sex biased perception. I also found differentially expressed transcription factors from this analysis. And then I also, the last thing I did was a sort of reverse analysis to pull out the least biased, but still highly expressed genes to try and identify candidate core olfactory receptors. I hypothesized that these are probably important for predatory activity. I also want to remark here that discovering details about sex bias and potentially mate detection were never my intention. However, I think understanding these dynamics is just as important, and I'd really like to investigate this more in the future. To take a break from all of this genetics talk, I just wanted to share this quick video of what I'm calling the sexy snake sniff, which is a male-specific behavior in snakes. So yeah, we'll just let this play. In this video, it's the male snake is actually the one on top, and he's smelling the female. This is probably a pretty laggy video to watch over Zoom, but hopefully you guys get the idea. It's just the male smelling the female. But I really like this video because it gives us a glimpse into the more gentle side of rattlesnakes. So yeah, it's only the males that we ever see doing this, as far as I'm aware. Let me know if people have seen the females do this. All right, so for this next section, I wanted to see if I could find evidence for genotypic integration between sensory perception and venom. I made this figure to show the position of every single venom gene found in the genome. The lighter boxes just magnify the green highlighted chromosome regions. On chromosome 2, I found what I predict are envenomate and relocate predatory loci. So let's take a closer look at that. If you zoom in, you can see two examples where toxin genes are directly flanking chemoreceptor genes. The first includes a paralysis-causing toxin called myotoxin, abbreviated here as MYO, and it's directly flanked by several V2R genes. Further down on chromosome 2, we see a different toxin which causes hemorrhaging and bleeding called L-amino acid oxidase. And it is directly flanked by olfactory receptors, including several that I marked earlier as core predatory candidates. So could this first region be for paralyzing and then tracking down prey close range? And could the second region be for inflicting and detecting hemorrhaging? I don't know for sure, but I would love to test this more in the future. Next up, I want to look at venom ontogeny. I'm going to take a little sip of water. All right, this is building off of previous work that was done by Mark Margris here at FSU, who determined that venom composition and function do in fact change as eastern diamondbacks shift from juveniles to adults. And this appears to be tied to shifts in prey preference. Here I'm just showing one of Mark's PCA plots highlighting this phenotypic shift. First, I confirmed that the old and the new gene accounts matched each other. Then I repeated the same differential expression pipeline that I described earlier, except this time I had 18 individuals spanning all size classes. From the expression tests, I again found age-biased venom genes along with transcription factors that were biased for age. Again, these heat plots are oriented smallest to largest, left to right, just like before. I know you probably can't read everything here, but I just wanted to show these so that you get a rough idea of how many genes we're talking about and how they are biased. So like this group at the top would be the adult biased venom genes, and the smaller group at the bottom would be the juvenile biased venom genes. And same thing over here, but these are the transcription factors. In total, I found 24 significantly biased venom genes. 18 of these were higher in adults and 6 were higher in juveniles. I also found 37 significantly biased transcription factor genes with 26 higher in adults and 11 higher in juveniles. Several of these differentially expressed transcription factors, including one called BH-LHE40, were biased mirroring the olfactory bulb findings, suggesting that these may, in fact, be global age regulators. Now I'm going to dig much deeper into the world of regulatory genetics and share some of my findings from this past year. This work has been setting up a regulatory framework for venom, and even though I'll be focusing on venom for the rest of my talk, this framework will set the stage for future work on sensory perception. We performed ATAC-seq and cut and run on venom gland tissue to evaluate chromatin accessibility, which can tell us a lot about how gene expression is regulated. ATAC-seq relies on a TN5 transposase and is not picky about which open chromatin regions it targets. Cut and run relies on a defined antibody target and is pickier than ATAC-seq. We used the H3K27 acetylation antibody to target active enhancers. Similar to the differential expression tests I performed earlier on RNA, I used these sequencing reads to evaluate differentially accessible regions of the chromatin based on age. So prepare yourself for more heat plots. First up is ATAC-seq. The orientations are consistent with before, with SVL going from small to large. And now, instead of genes, we are actually looking at sequencing peaks, which were called using the MAX pipeline. So these are actually much smaller than genes. They're just small areas where we were able to capture these signals. Here I split differentially accessible hits into 4 heat plots based on where they occur in relation to venom genes. The first of these right here shows differentially accessible peaks inside of the actual gene region. Second, we have peaks flanking up to 1 kilobase, and then between 1 kilobase and 10 kilobase, and then finally between 10 kilobase and 100 kilobase. I won't go into much detail here, but the reason I split these up is because we expect to see different types of regulatory mechanisms based on their spatial relation with the gene. For example, the promoter elements influencing differential expression would probably be captured in either the first or the second plot here. All of this was again repeated for cut and run. As you can see, we had much lower sampling for the cut and run test. We mainly just wanted to confirm that our findings were consistent between both ATAC and cut and run. I think this is a much easier way to visualize everything that I just showed you, with the RNA, ATAC, and cut and run now all stacked and colored according to their biases. Basically, we see agreement between accessibility and expression bias, which confirms that changes to chromatin structure is at least partially responsible for changes in gene expression. And so, for example, we see blue, 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 red, red, red. So all that means is that those biases are matching. From here on out, I will focus on a group of toxins called snake venom metalloproteases, or SVMPs. These are the same ones that I just showed you in the last figure, and they're all found on chromosome 9. SVMPs actually make up a large proportion of the eastern diamondback's venom. So continuing my goal of establishing a regulatory framework, from my earlier analysis, I found candidate transcription factors that might be responsible for changing chromatin structure or otherwise influencing transcription. One of these is a known repressor called BHLHE40, and is upregulated in adult diamondbacks based on the RNA expression. So I hypothesized that this transcription factor represses the juvenile biased genes in the adults, making the signal appear comparably larger in the juveniles. I was able to predict a DNA binding motif that this transcription factor will bind to, which then I used to perform an enrichment analysis. And I found that this motif was indeed enriched around the juvenile biased genes. I decided that my last step was to compare this finding with another species that is not known to have a functional ontogenetic venom shift. And this would be the Western diamondback rattlesnake. And before I show you those results, I wanted to share this protein-protein interaction map for 3 different snake species. Here we see that BHLHE40 interacts with proteins related to circadian rhythm and other timing related functions. Literally, one of these proteins is just called clock. And you can see all 3 of them have it. The only reason I chose these 3 species are those were the ones that were on the database that I used. To orient us to this, we are now looking at the same SVMP gene array as before, except it's now aligned between the eastern and the western diamondback with the biased genes colored blue and red, the same as before. And the motif hits for BHLHE40 are colored green. And I actually included the actual motif down here. So anywhere in the genome for this region that we see this, I put a little green blip. So that's what we're looking at. So now I want to zoom in on these 3 juvenile biased eastern diamondback genes. Only one of these is biased in the western diamondback. So now we're zoomed in. And now we can see 3 additional BHLHE40 motifs for the eastern diamondback compared with the western. And so they're right here. So now we can single one of these out and zoom in again. And finally, we can see a clear story where a couple mutations generated changes to the BH-LHE40 binding affinity between the eastern and the western diamondback at this location. The expression tests from earlier told us that adult eastern diamondbacks produce more of this transcription factor than the juveniles. So when BH-LHE40 sticks to this location, it likely somehow switches off the nearby SVMP genes in the adults. For my last takeaway message, I wanted to ask if anyone here has ever heard of the term junk DNA, or maybe they learned somewhere that a large proportion of the genome has no known biological function. Basically, by identifying this binding site in rattlesnakes, it's a perfect example where a genome sequence presumed to be junk DNA now has strong support for actually being a regulatory element influencing ontogenetic shifts in venom composition. And with that, I'll close with a recap of what I've gone over today. So what are the genes that allow rattlesnakes to detect their prey? And these are the olfactory receptors and the type 2 vomeronasal receptors, and the other ones that I went over at the beginning. What is the molecular evolutionary baseline for sensory perception? Olfactory receptors and V2R genes occur in large tandem gene arrays spread across the genome. Extracellular regions of these receptors are significantly more variable than the intracellular regions. And a V2R expansion occurred in snakes following the divergence from a lizard ancestor. And last up, is there genetic integration between sensory and venom repertoires? Is trait coevolution detected? First and foremost, I found strong evidence for physical linkage on chromosome 2. I also found differentially expressed olfactory receptors and toxins, and this suggests a shared ontogenetic driver. I found differentially expressed transcription factors with matching age bias in both tissues, including a repressor called BH-LHE40. And finally, motif enrichment supports BH-LHE40 as an SVMP ontogenetic regulator in the eastern diamondback genome. Looking forward, I hope to eventually reach a point where I can predict important ecological factors directly from predatory gene sequences alone. Doing so could provide valuable insight for the conservation of more vulnerable species, such as this incredibly rare transverse mountain rattlesnake. And finally, any progress untangling the mechanisms of gene regulation is directly relevant to ongoing research on things like cancer, gene therapy, and age-related illness. I hope that my contributions could someday become valuable in these areas as well. And with that, I would like to thank all of our funding sources that made all this research possible, including the National Science Foundation, Florida State University, and of course, the American Genetic Association. And I also would like to thank my lab, especially Darren, who has been just an incredible advisor over the years. And I hope to continue working with you. And yeah, I want to thank my grad committee, all of our wonderful collaborators, the King building admin, the custodial staff, especially Alex and Josh for their constant technical support and for maintaining all of our servers that I did all of these analyses on. Last but not least, I'd really like to thank Alexa and our cat Wesley for being my rock, and my always supportive family, and all my awesome friends. And all these people and memories have made grad school an absolute blast. So thank you all. And with that, I just want to thank everyone again for joining me today. And I think we have some time, so I'm happy to take any questions from people. All right. Thank you.
That was great. Yeah, Mike, I'll just let you call on people or people can unmute their questions.
I guess David.
Yeah, so you never talked about how much more expressed these genes are when they're, differentially expressed in adult and juvenile. Are they like turned off completely in one phase and then completely upregulated, or is it kind of a subtle thing?
It's different from one to the other, but it's relative expression. So like, for example, like on that, on these heat plots, like those, every gene is not expressed the same amount, but based on the total expression that we calculated, it's normalized between the two, if that makes sense. So,
Well, yeah, no, that doesn't really answer. I mean, is it a factor of 10? Is it a factor of 100? Is it a factor of 2? I mean, of course, it's not the same for every gene, but I mean, can you give me some sense of the actual magnitude of the differences?
I couldn't reliably, like, there's a I can tell you there's a huge range, and it could be like between like 2 and 5 orders of magnitude. So, but the thing is, like, for a toxin, it doesn't necessarily matter if there's just a few copies of it or there's thousands of copies of it. If it just, it depends on the toxicity and how they work together. So yeah, I could get you that information, but I don't have it just off the top of my head. But I am confident that that was included in the differential expression comparisons.
Sure, I don't doubt that. It's just like, it's just a question, is it subtle or is it like, really wacky over the head kind of thing?
I will tell you, as far as like the big functional shift in venom between the juveniles and the adults, the juveniles, it is mostly those SVMP genes that are making that big difference. And actually the highest expressed gene changes between the juvenile and the adult. So the highest expressed gene in the juvenile is one of those 3 that I showed you, the blue ones. And then it switches in the adult to a different gene, and those 3 get turned off. So, and like, yeah, like we're talking about like most of the expression in the venom gland, if that makes sense. Daniel?
Great talk, Mike. Thanks for the invite. Well worth getting up on Saturday morning.
Yeah, are you in Australia right now?
Yep. So
thanks for making it.
happy to be here. That was fantastic. Could you just give a little bit more detail about what like the advantage would be of having toxins and like the olfactory genes associated specifically with detecting the effects of those toxins so close to each other would be? Like why it, like, I get they're right next to each other, but what's the actual advantage of having such tight spatial linkage?
Yeah, that's a great question. And so there has been a little bit of work done on this where like, there's a certain effect from a toxin that produces an effect, like it kills the animal somehow. So like, it's either like causing like almost rotting of the tissue, or it's sending out a certain smell. And so like, if you can combine, or if you can keep the thing that makes that smell and the thing that detects that smell close together, then that would be very adaptive. Like, that would be like a good package to keep, like I'm just going to keep all that together and not spread it around where like, rearrangements of the genome or things could actually like break that connection, that, behavioral reliance from one to the other. So I don't actually, I really don't know like exactly what those things could be. I know I put like the 2 predictions of like, maybe it's this, but another example, like maybe there's a toxin that makes a mouse just like, urinate or something. And like you have the gene that detects that right next to the toxin that causes it. And so who knows, like that could be part of it too. So, but yeah, I think that by identifying these little clusters, like we can kind of hone in and be like, okay, like what do those things actually do? And like obviously a lot of work has been done on the venom, so we know what each of the toxins do, but now we can follow it up with the receptors and see, okay, well, what cues are they detecting, and try to kind of complete that story. So I don't know if that answered your question.
Yeah, thanks. That's very cool. Looking forward to seeing where that goes.
Sweet. Thanks for coming. All right. How do I? Other people in the chat? I don't know if anyone else has questions. I can't tell though. David, did you have another one?
Yeah, so what are the juveniles eating that's different from the adults? And can you connect that with any of these changes in venoms and receptor stuff?
I can tell you what the difference is. So like the difference in diet. And so a really massive rattlesnake is not going to waste its time eating very small mice. And so we see a big shift from the juveniles. Like if you think about it, like a baby rattlesnake is small, and a full-grown eastern diamondback can take full-size rabbits. And it makes more sense as they get bigger to not waste their energy tracking down all these small, fast-moving prey and just have a couple really big meals a year, and that's it. And so that and the functional support for that, Mark Margus did, was looking at the different LD50 values between the different prey sources. So I didn't do any of that work. All I did was try to build off of it. And yeah, I would say that connecting the receptors to their targets is a huge missing part of this. And there really isn't a way to like predict it without, really complicated like knockouts or like really sophisticated modeling. But I think that in the next couple of years, we will see a lot of advances that enable us to do that. And that's kind of what I'm looking forward to. Oh, Kim has a question.
Hi, Mike. Great talk. I
Thank
really
you.
enjoyed it. I had a question about your sex chromosome linkage of the sex biased genes.
Yeah,
That was a very cool result.
thank you.
I think a very large portion of those were male biased, and you had that nice video showing some of the behavioral differences. The genes on the Z chromosome are going to be present in females, but do you have any insight whether they're down regulated more than 50% compared to the males?
Yeah, so you're talking about compensation, dosage compensation?
Is it just that they only have females only have half as many copies, so they produce half as much, or have they differentially regulated the half number of copies that they have?
Yeah, that's a great question. And actually, Darren wanted me to put more in there, but there wasn't a whole lot of literature that I could find that gave a clear answer to that. And the best I could do is say, I found around 30 genes in the males that were biased, but there were way more than 30 genes on the Z chromosome. So why didn't I see all of them up 50% because the male had the second copy? And the other kind of argument against that is that I think there was one sex bias gene, and that was actually one sex bias gene for the female that was also on the Z chromosome. And also a lot of the core olfactory receptors I found, I think around half of them were also on the Z chromosome. So I found that there was exactly, very similar expression between the two sexes. Not enough for a significant signal, but they were all still on the Z chromosome. So I actually don't know what's going on. It just seems like there is some sort of sensory evolution that's going on with the Z chromosome that like there's just a huge amount of genes on there. Like those two donut plots I showed, it was almost half of both of them. So yeah, I guess I don't know, but
yeah,
it's really interesting.
It's very interesting. It does seem like there's lots of differential regulation. It's not just due to compensation. So very cool. Thank you.
Thank you.
That was great, Mike. I've been through the chat and it's just everyone saying how great your talk was. So unless someone has another question.
Yeah, thanks everyone.
Yeah, great job, Mike.
Yeah. Cool. Should we call it then, or?
Yeah, we'll call it. Thank you. That was great.
All right. Thanks everyone. Thanks for coming.